live streamed. We'll get started in a minute, literally max. Cool. Let's get right into it. So today's is basically the basics. When I say that this is the end of kind of the very basics of biology that we've been talking about for a couple of weeks, we've been talking about very simple processes. This is the core. It's cells and organelles. So things that are found in cells, all of the organelles, the functions of the organelles, how does a cell work? What's in a cell? And then we'll talk a little bit about microscopy and how to identify cell structures. So cell theory is the basic building blocks of biology. It's basically the idea that all organisms are made up of cells, and cells all have the same fundamental structure. So a cell is basically a building block. It's like a Lego brick, and then you make this larger organism built up of lots and lots of cells put together. And complex organisms, lots and lots of cells work together to make a tissue. Lots and lots of tissues come together to make an organ. Lots and lots of organs come together to make an organ system. And then all of these organ systems come together to make an organism as a whole. And these are really important basic biology to understand. A prokaryote is a single-celled organism which lacks a true nucleus. So it's really important to understand what a nucleus is. So a nucleus is the store of DNA in eukaryotic organisms, and prokaryotes will lack this true nucleus. Yeah, hello, Rana. Yes, this meeting is being recorded and it can be viewed on YouTube. So prokaryotes are single-celled organisms which lack a true nucleus, i.e. bacteria organisms. And they will contain different structures and different building blocks than eukaryotic organisms. And we'll talk about that in a little bit of detail. Eukaryotes are single-celled and multicellular organisms. So eukaryotes can be single-celled as well, but often they will be multicellular, larger organisms, which have a true membrane-bound nucleus. This is really important. It's a membrane-bound organelle, which basically means that the nucleus is surrounded by its own membrane. And that's really important because what that does is that protects the nucleus. It protects the genetic material inside. The membrane that binds the nucleus makes transcription essential because it means the DNA cannot leave the nucleus and cannot be directly replicated into proteins. It has to be converted into mRNA first because it means that the DNA is too large to leave through the nucleus. A membrane-bound organelle is another key term. It's an organelle that is separated from the cytoplasm by its own membrane. Can you guys in the chat, work together to identify all of the membrane-bound organelles in a eukaryotic cell? Can you hit me with all of the membrane-bound organelles? Nucleus, yeah, is an important one. Ribosomes and chloroplasts, both of them are important. And this links back to endosymbiotic theory, which is the idea that ribosomes and chloroplasts both were separate bacteria that were engulfed by eukaryotic cells and became part of that. Mitochondria, yes, is separated by an inner and outer membrane. The Golgi is, yes. Rough endoplasmic reticulum will, yes, and so will the smooth. They're both separated by membranes. The name of the theory that I just mentioned was endosymbiotic theory. It's really important to note. It is something that comes up in some specifications. The vacuole is membrane bound, yeah. And animal, plant, algal, and fungal cells are all eukaryotic. And this is a really good summary table. And we'll talk about this in a bit more depth as we go into it. But we'll talk about all of these different structures. And it's really important. Maybe you might want to take a screenshot or some sort of note of this table. This is a good summary, just showing you what organelles different eukaryotic cells will have. And we'll talk about the differences between them as we get further along. Give it a second to take that down. Okay. No. Anything that isn't membrane bound? Good question. So the cytoplasm obviously isn't membrane bound. The plasma membrane is its own membrane. The cell wall won't be membrane bound. Ribosomes aren't membrane bound. And there's the big ones that aren't membrane bound. Lysosomes are membrane bound. Yeah, lysosomes are membrane bound because they have a membrane that protects the hydrolytic enzymes from getting out of them. The cytoplasm is membrane bound though. Yes, technically, I guess from a technicalities perspective, the cytoplasm is bound by its own, the plasma membrane. Rough endoplasmic reticulum, no, that is membrane bound. The endoplasmic reticulums are both membrane bound. Organelles are substructures within cells. Every organelle within a cell has a special function that contributes to the overall functioning of the cell. And we'll talk about these kind of as we go. We're going to talk about these now. So, starting with the nucleus and ribosomes. So, nucleus is made up of so the nucleus is this overall shape. It's this overall spherical shape in the middle of the cell. It's made up of a nucleolus, which is surrounded by chromatin. There's a nucleoplasm, which is like a cytoplasm, but for the nucleus. It's a reaction medium, but only for the nucleus. And is surrounded by a nuclear envelope. This nuclear envelope is the membrane that binds the nucleus and separates it from other structures outside. The nuclear pore lies between within the nuclear envelope, and that allows for the exit of mRNA, but not DNA, which allows for transcription to occur. So all eukaryotic cells have a nucleus, and the DNA is packaged into as chromatin into the chromosomes. These chromosomes are stored at the center of the nucleus. The nucleus has its own double membrane called the nuclear envelope. This regulates what gets into and out of the nucleus through nuclear pores, which are found throughout the nuclear membrane. The nucleolus is the nuclear region where ribosomal RNA is assembled into ribosomes. There may be multiple nucleoli. And the nucleus's function is to control all of the activities of the cell, as well as controlling how the cell interacts with its external environment. It regulates the two processes of DNA replication and DNA transcription. And we've talked about both of these before in previous lessons, but transcription, to summarize, is where the genetic material that is stored as DNA is turned into RNA, which is made into the protein by ribosomes. And no, so good question here from Shaden. Does transcription happen in the nucleolus? No, it doesn't. It happens in the nucleoplasm, but the activated RNA molecules that come in and are used in the process of transcription, they come from the nucleolus. So what's really important to note is that those activated RNA will come from the nucleolus, but transcription itself happens in the nucleoplasm. The last other structure we need to talk about is ribosomes. So ribosomes are small organelles with two subunits. A ribosome has a large and a small subunit. Eukaryotes have ATS ribosomes, which, as I mentioned in a previous lesson, is made up of a 6DS and a 40S ribosome. Is anyone able to comment and think for a second here, why, when we have a 60S and a 40S ribosome, why that doesn't mean that the ribosome, why that means that the ribosome is in 100S, because that obviously doesn't add up. Can anyone think? If you're able to take a moment, maybe pause and think about it. What is the 20 goes away somewhere, says Eliza? Not where does it go? What is the S? The subunits form together, fuse together, some denature. Interesting thoughts? Not exactly. Density, you're getting closer, Amy. Well done. Size, yeah, sort of getting in the right, we're in the right ballpark now. Not merging, it's not exactly to do with merging. Where it goes when centrifuged. Amy, you're on the right lines. Yeah, it's basically to do with sedimentation rate, which basically means that the S is actually something called a Svalbard unit, which it's like a spelled really badly, but it's the sedimentation rate, which is basically the amount of time it takes for the organelle, if you put it in a centrifuge, for the amount of time for it to settle at the bottom in like microseconds, in like a very small, it's like 1 × 10⁻13 of a second. So this is very small units we're talking about. And an ATS and a 7, and so each of these subunits have different rates, but as a combined species, they all have a bigger sedimentation rate, but actually not enough to be the combined sedimentation rate of each of the subunits. What is a centrifuge? Asks Mariam. Good question. So a centrifuge is a machine. Basically, you get a machine. I'll kind of draw it out. So you've got this central machine with different arms, and you put a test tube at the end of you put your test tube with your sample. And what the centrifuge does is it basically spins this very, very fast. And by spinning it very, very fast, what you're doing is you're separating the lighter structures from the heavier structures. Because what's happening is the speed at which, remember, speed is to do with the amount of kinetic energy you have is to do with 1/2 times the mass times your velocity squared, right? You remember that from physics. Your mass is really important because your velocity is constant. Your velocity depends on the amount of the rate of spinning of the centrifuge. So this is to do with the rate of spinning. So your mass is what determines what your kinetic energy is going to be. And so the larger the kinetic energy, the more likely you are to sort of come towards the top, and the smaller your kinetic energy, the more likely you are to sink towards the bottom. And that's why it is really important to note that. Hope that makes sense. Do we need to know how a centrifuge works? Asks Ashar. No, you don't. You don't need to know how a centrifuge works. No worries. I was just explaining it for some general interest. Ribosomes are mostly found freely floating in the cytoplasm, but some can be bound to the endoplasmic reticulum, and specifically the rough endoplasmic reticulum. And actually, the type of protein that you're forming depends on whether your protein is formed in the it depends on whether the protein that you're forming is going to be formed on the free ribosomes or on the ribosomes that are bound to the rough endoplasmic reticulum. We're not really going to need to talk about that. This has been a question I've got a question here. I can't see the name, unfortunately. How do we see the difference between the rough endoplasmic reticulum and the smooth endoplasmic reticulum when it comes to labeling? A really good question. So you see this cell? That's your Golgi. Your smooth endoplasmic reticulum doesn't have the bubbles. The rough endoplasmic reticulum is the side with the balls on it. And that's usually when it comes to labeling. If you're labeling an actual cell diagram, you won't be expected to label the difference between rough and smooth endoplasmic reticulum, as long as you're able to identify that it is just the endoplasmic reticulum, purely because on a light microscope image, you're never going to be able to see the ribosomes. So you'll never be able to identify it. But ribosomes are made up of rRNA and proteins, hence they're a type of ribonucleoprotein, and they read the mRNA and use the mRNA to make proteins. That's their job. Okay. We're now going to talk about the mitochondria. So, the structure and function of the mitochondria. So let's zoom into the mitochondria. So the mitochondria is made up of an inner and outer membrane, and the inner membrane is folded into cristae, and these cristae help increase the surface area for the ATP synthase molecules that are used as part of respiration. And you will learn a lot more about respiration in A2. And we'll talk a lot more about that in A2. But for now, let's go with the assumption that you don't need to know that much about it, but you need to know that the inner membrane, so it's a bilayered, it's a bimembranous structure, it's got an inner and an outer membrane. Inner membrane folded into cristae, outer membrane forms the capsule. The matrix is like the cytoplasm of the mitochondria. It is a reaction medium and it contains enzymes that are used for respiratory processes. Ribosomes are used in the mitochondria to form mitochondrial proteins that will be useful for the enzymes that, because if you imagine, then really nothing big, like no proteins can get into the mitochondria because it's a membrane bound organelle, which means that the proteins that the mitochondria needs has to be produced from within the mitochondria itself. It's why the mitochondria is the only other organelle bar the nucleus to have its own DNA. And it means, and you inherit the DNA of the mitochondria from the, you inherit the mitochondria from you inherit your mitochondrial DNA from your mother. And that's really important to know, but something that's quite interesting. Question from Yusuf. Sorry, are the dots around the rough endoplasmic reticulum ribosomes? Yes, they are. But yeah, so you've got the matrix, it's got the DNA in it. You've got ribosomes to produce proteins that are specially used for the respiration process itself, which means that any disorders of the mitochondrial DNA usually present themselves as disorders of the respiration process. Sorry, apologies about that. But yeah, usually the clinical presentations of patients with mitochondrial diseases are problems with respiration, and they usually can present themselves in loads and loads of different ways, but they're quite interesting as a range of conditions. I've had a really good question here from Hanan. Why do we only inherit our mitochondrial DNA from our mother? And this is because basically, if you imagine what's actually happening when you're fertilizing an egg, you've got the egg, you've got the sperm, not to scale. The sperm will infiltrate the egg's outer membrane, but the sperm itself is actually degenerates. The only thing that the sperm releases is its genetic material. And so when the sperm releases its genetic material, it only releases the 23 chromosomes. So the mitochondria that's still left are the mitochondria left from the egg cell itself, because the sperm cell degenerates. We've had a good question here from Ali. Do we need to know about mother inheritance thing for AQA? Yes, you do. Not at AS, not in this topic particularly, but I like to integrate slowly the different processes throughout. So I will integrate content from a little bit more advanced down, just so you can get a bit of understanding on it earlier, but you will need to know it by A2. But yeah, as I said, inner membrane is folded to create cristae. Inner region is called the matrix. It's like a cytoplasm. And as the old phrase goes, the mitochondria is the powerhouse of the cell. And it's the site where aerobic cellular respiration and ATP synthesis occurs. The mitochondrial matrix contains enzymes that are necessary for cellular respiration. Mitochondria have its own DNA, as we talked about that earlier, and they have their own 70S ribosomes. And actually, interesting fact, because you do need to know about endosymbiotic theory, and so I'll give a little bit more talk on that. The 70S ribosomes are the ribosomes that you find in prokaryotic cells. And this was actually the initial evidence that indicated that ribosomes were engulfed by, mitochondria were engulfed by animal cells millions of years ago in our evolutionary process, because they discovered that ribosomes have 70S ribosomes, mitochondria has 70S ribosomes, whereas normal eukaryotic cells will have 80S ribosomes. So this is actually a part of the evidence for endosymbiotic theory. And the cristae help to enable compartmentalization. Does anyone know the definition of the term compartmentalization? You've reached about October in your A-level process, so you should have heard of it by now. A lot of the AS people and the A2 people. I expect both of you to have heard of this. What is compartmentalization? Yeah. So compartmentalization is separating the cellular compartments into different sections of the cell. Yeah. So it basically means that the different stages of the respiration process happens in different places. It helps protect the cell, and it helps protect the internal environment to be specific for the requirements of that process. For example, some processes work better in acidic environments, whereas some processes work better in alkali environments. Okay. Cell membranes and cytoskeletal structures. I'm going to quickly stop for a second here. If possible, again, as we did sort of in our last session, if it would be possible for you guys to leave a review for the class, I know it would be incredibly useful for us. And we have said that if you leave a review and you leave your email in the chat, we'll be giving away 1 free hour of one-to-one tutoring to a random person who has left a review for us just from this class. It would be incredibly useful for us, just helps get us out there. I'll stop for a couple of seconds. I will go over how compartmentalization works. Yeah, if you write your emails in the chat, and then and once you've filled out the review, then we can compare across, and we'll be giving away 1 free hour of tutoring. And I will go over how compartmentalization works when we talk about plasma membranes, which is now. I'll stop for a couple of minutes, just give you some time to write a review, and then I'll carry on.
Sorry, do we just put our emails in the chat?
Yeah, emails in the chat once you're done. I will wait another minute. Right, let's get back to the lesson at hand. Structure and function of the cell membrane. So the cell membrane is made up of two layers. So it's a bilayered, and it's a phospholipid structure. So we've talked about phospholipids in a previous lesson when we talked about the structure of them. It's composed of a phospholipid bilayer with proteins regularly embedded across its length. And the function of a cell membrane is to provide structure to the cell, but also to allow for selective entry of structures in and out of the cell. A cell membrane has lipids and carbohydrates attached, which have their own specialized functions. And again, so the function of the cell membrane is to regulate movement of substances into and out of the cell. And it overall acts as a barrier to protect the cell from changes in the external environment. But you can see that there's loads of different structures throughout it, and we'll go into this a lot more depth. Cell-to-cell communication happens through the cell membrane as well. There are special proteins called receptors, which are forms of glycoprotein and glycolipids, which receive signals, i.e. hormones or other receptor molecules, so other lipid-soluble hormones, from neighboring cells or from the environment. They then relay these chemical messengers within the cell to initiate specific responses. The other really important structure to talk about is the cytoskeleton. The cytoskeleton is made up of protein filaments, which form a mesh-like network in the cytoplasm of cells. These are present in all cells, including bacteria, and they're formed of both microtubules and microfilaments. So microtubules are basically, if you imagine like the train tracks, right? And they work in a sort of a 9, 3, 4, 5, 6, 7, 8, 9. And then, so you've got like a 9+2 structure for microtubules, and they're cylindrical proteins, and they're like the train tracks for things to move along them. And microfilaments are protein strands. So they're thin strands of proteins, and they're used for anchoring. They're used for like pulling things along. The cytoskeleton forms a range of functions. It strengthens and maintains the structure of the cell. It provides support to cellular organelles in maintaining their position. It's involved in the movement of cellular material, e.g. moving chromosomes through cell division. And it's also used for mobilizing the cell, so keeping the cell kind of when the cell wants to move through the flagellum. The flagella is made up of microtubules. Again, specialized cytoskeletal structures, rather than just acting as protein filaments in the cytoskeleton, these project from the cell, and they're cilia and flagella, and they're used for the motion and the movement. Centrioles are made up of microtubules, which are small, hollow, cylindrical proteins. They're found in most animal cells. They are found in most animal cells, but are only present in certain plants. Centrioles help chromosomes separate during cell division by pulling the chromosomes apart. The centrioles also produce spindle fibers, Spindle fibers are what actively does this process. Cilia are hair-like structures on surface membranes. There are motile and non-motile cilia. Motile cilia can help move due to the activity of the microtubules inside it. This type of cilia helps move substances along the surface of cell membranes, i.e. respiratory or digestive cilia. Non-motive cilia do not move, and they're found in sensory organs, like in the eyes, in the photoreceptor cells of the eyes. And what they do is they project outwards. A lot of bacteria also have non-motile cilia that project from their structure, and this is used for transfer of genetic material between bacterial cells. Really important in the production of antimicrobial resistance. You'll learn about this a lot more in A2. Are there certain cytoskeletal structures that we need to know apart from flagella? Well, cilia, you need to know about centrioles, you need to know about microtubules and microfilaments. The flagella are longer protrusions from the cell surface. They're thicker and longer than cilia, and they're covered in a layer of the plasma membrane itself. They also have microtubules inside it, but these are purely motile. They're only used for moving the cell and propelling the cell through a structure. For example, the flagella at the end of the sperm tail. I've been asked a question about Shreya. Are there going to be separate webinars for A2, or will these lessons going to continue with year 2 content? The plan is hopefully to continue these lessons after Christmas with year 2 content. So hopefully. Which cells other than bacteria and sperm are flagella found in? They're found in a very high range of cells. Think about so many different cells. My mind's going blank. Fungal cells, they're found on fungal cells. I mean, that's the easiest cop-out option. But flagella are found in loads and loads of other cells in humans as well. If you think any cell that really needs to move, Golgi apparatus and lysosomes. So we're not going to go into this in too much depth, actually. The Golgi has a cis and trans phase, but the function of it is to package lipids and proteins to be exported outside the cell or to its surface membrane. That's the function of it. And there are 2 faces. So the cis face, which is the front facing, that receives proteins and organelles, and the trans face releases. So that's the one towards the plasma membrane, and that releases the proteins after packaging. And the middle bit, which is called the cisternae, that does the packaging itself. You don't need to know this in any detail. The Golgi apparatus also makes lysosomes, and we'll now move on to lysosomes quite quickly. Lysosomes are round membrane-bound organelles, no distinct internal structure, and they're an offshoot of the Golgi apparatus. They contain lysozyme enzymes. Lysozyme enzymes are hydrolytic enzymes, which means they do the process of hydrolysis, and the function of these is to digest material either from within the cell or from out with that's dangerous. Again, the membrane surrounding the lysosome protects the rest of the cell from these very strongly digestive activity. Lysosomes are hydrolytic enzymes, so they're used for digesting organelles, digesting bad bacteria, and worn out structures within the cell. But again, you don't need to know this in too much depth, and we'll go into this in a lot more depth later. Do we need to know about exocytosis? Yes, but later. Not right now, not in this lesson particularly. Probably in the lesson when we do plasma membranes, I'll talk about that. Endoplasmic reticulum. You do kind of need to know about this. So rough endoplasmic reticulum, stacks of thin fluid-filled membranes. It's a membrane-bound organelle located near the nucleus. A rough endoplasmic reticulum is covered in ribosomes, and the ribosomes make proteins that are exported outside the cell or for the cell membrane. So if you think going out of the cell, those proteins are made on the rough endoplasmic reticulum. Proteins that are used inside the cell are made on free-floating ribosomes. Really important to note that. The rough endoplasmic reticulum folds and packages proteins, which are made by ribosomes attached to the rough endoplasmic reticulum, and sends them to the Golgi for export. Kind of simple. And then smooth. So the smooth endoplasmic reticulum doesn't have ribosomes, hence smooth. And its function is to synthesize lipids, i.e. cholesterol, and other molecules, i.e. steroids and hormones, for intracellular use and transport. How can you tell the difference between, you won't. This is a question I have been asked earlier. Telling the difference between Golgi and, Golgi and SIR in a cell diagram. So they do look completely different. So SIR looks like basically thin stacks, basically. Whereas the Golgi, from a front-on perspective, is just going to be lines. So it's completely different. It's just about this, the function. Ghazal, does the SER also detoxify harmful substances? Yes, but not as commonly. Its main function is synthesis of lipids, and that's the function you should be writing for your exams. And again, when you make lipids, you go to the Golgi for export. The SER also plays a crucial role in detoxification that has been consumed by the cell, i.e. alcohol. But again, don't need to know this in too much depth at the moment. This is definitely something that you'll come onto when you do a degree. That this is beyond, this is university level content. To understand that. Isn't the Golgi will have lysosomes around it on a diagram? But to be fair, a good point, Jess, but actually it's highly likely that the smooth endoplasmic reticulum will also have lysosomes around it on a diagram. If it's an actual cell diagram, yes. Okay, but what's important is you need to know what the positioning of these will be. So smooth endoplasmic reticulum, you'll see that it kind of looks like one continuous piece. It's like a snake, whereas the Golgi looks like separate faces because they've been cut in a transverse plane. So it looks very much like 5 different unjoined stacks. The only thing I need to talk about when it comes to plant organelles is chloroplast, because that's the only thing that's different, as well as the cell wall. So chloroplasts are made up of structures of grana. Grana are stacks. So a granum is different stacks, is a stack of thylakoid. Thylakoid is a very similar structure to like the cristae, and it's the thylakoid membrane that so chloroplasts are key for photosynthesis, found in algae and plants. Again, very similar. They follow the same endosymbiotic theory. So they have small DNA and 70S ribosomes. They're flattened membrane-bound organelles. They have thylakoid membranes, which are small and thin, and they're stacked up into grana, which increase the surface area of the thylakoid. The grana are connected to each other by thin pieces of membrane called lamellae. And stroma is like the matrix of the chloroplast. It's a thick, viscous fluid that fills up most of the space of the chloroplast. I'm not going to talk about this in too much depth because there's no point, because you will learn this in A2. What is the advantage of them being joint? So this is a good question. The advantage of them being joint is it means that things can pass between them. If a UV hits one thylakoid, the UV energy can be passed throughout so that the whole cell can produce glucose. Cell walls. So cell walls are found in, yes, good question, Kartika. Yes, lysosomes are involved in apoptosis. They're also involved in necrosis. So they're involved in both programmed and unprogrammed cell death, but that's kind of their entire function. So plant cell walls are a very thick structure made up of cellulose. In fungal cells, it's made up of chitin, and bacterial cells, it's made up of peptidoglycan. And this leads on to your question, Saeed. Yes, you do need to know what each cell wall is made up of. So you need to know cellulose, which is a polysaccharide of glucose, which we talked about earlier in a previous lesson. Chitin is very similar. It's a glycoprotein, and peptidoglycan, it's in the name, peptido, protein, glycan. It's a glycoprotein as well, glycoprotein structure. Peptidoglycan is for bacterial cells. Chitin is for fungal cells, and cellulose is for plant cells. The main function of the cell wall is to support a cell. It gives the cell structure, preventing it from changing shape, and the cell wall should be freely permeable. So anything can get in past the cell wall, anything soluble anyway, but the plants still have a cell membrane, which allows for selective permeability. And then the last thing is the large permanent vacuole. So again, plant cells have large permanent vacuoles, which are surrounded by a membrane called a tonoplast. And this contains, it's kind of like a sponge, is the way I would think about it. It's kind of like a sponge. So what it does is it absorbs water at times when there is high water potential in the cell, which means that it prevents the cell from bursting. And when there's low water potential, it will release some of the water in its sap and its chemicals. And again, this will prevent the cell from wilting. So it's basically a bit of a buffer for a plant cell before it bursts due to high water potential. Do all prokaryotes have peptidoglycan cell walls? No, not all of them. Some bacteria do though. Most of the ones that you'll need to talk about will. The vacuole isolates harmful chemicals from the rest of the plant. And the tonoplast separates the contents of the vacuole from the cell cytoplasm. That's kind of its function. Oh, I'm not going to talk about this because this we'll talk about later on. There will be another lesson on this entirely by itself. Okay, we've talked about this. This is important. So this will probably be the last thing we are able to cover just with time, because I do still want to give time for questions at the end. Cell specialization. So remember, cells have particular functions. So they have to go and do certain things. But all the cells have generally the same structure, which means cells differentiate to perform specific functions. You need to know about 3 cells and their specialization. So sperm, nerve, and red blood cells. And these are just examples of specialized cells. Remember, specialized cells will have different features that make them able to perform their function. So they might have more organelles. So sperm, for example, has lots of mitochondria, which is able to create, to release the energy that the sperm needs to be able to swim. Nerve cells have lots of ion channels, which allows for depolarization and allows for sodium ions to flow. And red blood cells have no nucleus, which increases the surface area for cell movement. So red blood cells, enucleated, which means that they have more space for heme, which means that they can carry more oxygen around the body. They also have no DNA and do not carry out, but they do carry out most other typical cell functions. Neutrophils, they have lots and lots of lysosomes because their entire function is to break down foreign material. So they're going to have a large number of lysosomes. Neutrophils also have a flexible shape, which means that it can mold and it can swim around other structures that are present in the blood. It also has a much higher amount of lysosomes, yeah. Epithelial cells, they have mobile cilia, which allows movement through the respiratory tract. Other epithelial cells, like squamous epithelial cells, are very flat, which means that gas exchange can happen efficiently in the alveoli, for example. Sperm cells, they have lots and lots of mitochondria. And this means that they can release energy, which means that the flagella can move and the sperm can swim. They also have something called an acrosome. So an acrosome contains enzymes that allow them to break the thick wall down, the thick wall of the egg cell. They have a big nucleus, which makes it easier to transfer genetic material, and a long tail for good swimming. Palisade cells are the primary site of photosynthesis. They have lots and lots of chloroplast because they do lots and lots of photosynthesis. Root hair cells have this large protrusion, which means that they have a large surface area, as well as a large permanent vacuole to increase the rate of osmosis, because the function of the root hair cell is to absorb water from the soil. Guard cells, their function is to protect the stomata. So they're around the stomata and they can open and close, which means that they also have large vacuoles to absorb water. But what they also have is they have thick inner cell walls, which means that they can, if they become rigid, they actually change shape because they need to change shape because they need to be able to open and close the stomata. When cell specialization, how does it occur? One piece of advice for an exam though, if they ever give you a type of cell that you're unfamiliar with and they ask you about the specializations of it, the best way to do this is to think about the function of the cell. So start by thinking, what does the cell need to do? Then think, what would it logically need to be able to do that very well? Let's say you're given a cell, let's say you're given a reticuloendothelial cell of the liver. You've never heard of it, but it tells you its function is storage. What is it going to have? If its function is storage, it's going to have lots and lots of ribosomes to make proteins that are used for storage. It's going to have a large Golgi to package things into vacuoles. It's going to have a large surface area, so it's going to have a wide cell membrane. Again, so that's when you think, when you start getting given cells that you're unfamiliar with, you need to start thinking from function to specialization. Cell organization. So cells with a similar function form a tissue. Tissues come together to make an organ. So there'll be like epithelial tissue, there'll be glandular tissue, there'll be muscular tissue, and a bit of nervous tissue, and it all comes together to form an organ like the stomach, for example. And different organs with similar functions come together to form an organ system. So the digestive system, for example, comprises of the mouth all the way down. Mouth, stomach, pancreas, and all of these perform the overall function of digestion. And humans have loads and loads of different organ systems that do similar, related functions as an overall process. Right, I'm opening up to just maybe one or two questions if anyone has any. Good question here I've got from someone. Why is it good for nerve cells to have lots of ion channels? It's basically so that depolarization can happen throughout the membrane. So it means that you can transmit an electrical impulse the large distance of the nerve, and you don't have to depolarize at every point. But again, that's a little bit for a bit more advanced content. Okay, what is the tonoplast made up of? It's a cell membrane, so it's made up of membrane organelles. All right, I'm going to wrap up. Can I go over compartmentalization? I'll go over it a little bit more in our plasma membrane session, if that's okay. Again, it's more linked to that topic anyway, which is why I think it would be more useful if I go over it sort of in that slightly different lesson. I'm going to wrap up here.